So when we talk about the various processes of this biotechnology or recombinant DNA technology, in that we see the first thing that we have to do in order to create a recombinant DNA is to isolate the gene of interest or isolate the genetic material, because that is the most important thing. Because if you don't have the gene of interest, how are you going to carry forward your whole entire work? Then the gene of interest that is there, it has to be cut at specific locations with the help of the restriction enzyme. Later on, the cut DNA or whatever fragment or gene of interest that you have to separate it and isolate it using a desired, you can say, method, that is your gel electrophoresis, which employs the separation of the fragmented DNA depending upon the electric field, right? Under the electric field, and that varies accordingly alongside the charge of the DNA and the size of it. Particularly, it is based on the size. Smaller one will go faster or farther, and the one that is bigger or heavier, it will stay behind. Then, once we are done separating, we take that gene of interest and then we make multiple copies out of it using the method called as PCR. Now, the amplification of that gene of interest using that PCR is going to, you can say, like if you have a single piece of gene of interest or two pieces, then that's going to create billions of pieces for you so that you can do as many, like readings and experiments at a time. And then the DNA fragment, the gene of interest, it has to be joined onto the vector. And we know that the vector and the DNA fragment or the gene of interest, it has to be cut with the same restriction enzyme. Then only it will get, you can say, then only it will get ligated. So we use DNA ligase for the ligation process. Then transferring of the recombinant DNA into the host cell has to be done, which we call as transformation. Now, once you're done making the DNA fragment and the vector together, what you've created is a recombinant DNA molecule. So now, furthermore, after transferring it into the host cell, we have to culture the host cell so as to produce that in a huge number. And we know naturally as the host cell will divide, so will the recombinant DNA. And then once all that is done, we extract the desired product and then we sell it in the market. Is it clear?
Yes.
So these are the various steps. So the step first is basically extracting the gene of interest. Now, you know that the DNA is present inside the nucleus mostly, or the nucleoid region if it is a bacteria that you're taking it out from. Now, for that, you have to, if it is a plant cell, if it is a bacterial cell, you have to degrade the cell wall. Then you'll go into the plasma membrane. Then you'll extract that DNA. But apart from that, the DNA is in a mixture. So it's, it's like combined with RNA, it's combined with protein and all of that. So you have to remove all of those in order to get the purest form of DNA. And that is done by, if it is a, you can say, a bacterial cell wall that you want to degrade, you will use lysozyme enzyme. If it is a plant cell wall, you will use cellulase enzyme. So by that, you have released the DNA. Now, since I told you that DNA is not present alone, it, like the other substances, they also have to be removed. So like if you want to remove the RNA, you use RNase, you use protease to remove the protein component. And now the solution only has the DNA present in it. And now the DNA, you can take it out by spooling or treating it with chilled ethanol. Now, furthermore, the fragmentation is done using the molecular scissors that we call as restriction enzymes, right? And then the digestion of that is done. But how do you know that the digestion is done? You have to check that by doing the gel electrophoresis. Now we know that in the gel electrophoresis, DNA being negative charge itself, it is loaded onto the negative pole of that electrophoresis chamber, and then under the influence of the electric field, it travels towards the positive charge. And this traveling depends upon the size of the DNA fragment that is there. Okay, and apart from that, now once the DNA reaches the end, in the mixture only we've added Ethereum bromide so that under the UV light we can see it, clearly visualize it. If not, we will not be able to see the DNA fragments moving anywhere. Now, once they've reached the last side, then what we do? We cut the DNA out of that. Now, we have the gene of interest that has been fragmented. Now that fragmented DNA is isolated and that step is called as elution basically, or extracting it from that agarose gel which is extracted from the seaweed. Now the gene of interest that you're taking out, you have to amplify it. Now another technique is PCR. Now you know PCR stands for polymerase chain reaction, which was developed by Carey Mullis. Now in the PCR, there are 3 steps that has to be taken: denaturing, right? Denaturation, annealing, and extinction. So basically, and in this process, you require DNA polymerase for the extension. You require, and see, the DNA polymerase that we use is stack polymerase because we know that, in this process, a lot of high temperatures are utilized. So in order to cope with the polymers, we know that the enzymes, they get denatured at higher temperature. So the stack polymerase enzyme, it does not get denatured because it is a temperature that is found in those bacteria such as Thermus aquaticus that live in the high temperature, like up to 95 degrees Celsius. Imagine that high. Apart from that, we require primer, we require DNTPs, ions, and salts. All of those things are required. Now, during the process of denaturation, the double-stranded DNA is made two single-stranded DNA. Like you separate, you break the hydrogen bonds and it is separated. And this is done at 94 degrees Celsius. After that, there comes annealing. Now, annealing is that process wherein the reaction mixture is brought back to normal temperature, lower temperature, and then primers are added during that. And in the last step, the extension part is where the primers are, you can say, extended so as to form the new sequence or the new complementary sequence of the DNA. This step can be done n number of times depending upon however many products you want or however many cycles you want it to run. So that is what polymerase chain reaction is, right? So always remember 2 to the power n is how you find out the number of, you can say, molecules that will be generated. And that n is the number of cycles that you're feeding into the machine. If it is 10 cycles, you'll do 10 to the power n. If it is 20 cycles, you'll do 2 to the, this thing, 2 to the power 20. So 2 to the power n, wherein n is the number of cycles that there are. Now, this particular PCR is done. You've, created a lot of copies. Now, you're going to take that gene of interest, we'll take the vector, cut the vector at the specific restriction sites, and then you load on the vector onto the restriction site using the enzyme called as DNA ligase. DNA ligase is the molecular glue, so it joins them two together. And now what you have is a recombinant DNA. Now this recombinant DNA, it has to be sent inside the host, right? Now the host cell has a cell membrane and cell membrane doesn't let anything come inside. So it has to be made competent. And in order to make the host cell competent, there are various techniques, there are various processes that we take into play in order to make the host cell competent. What are they? First is the heat shock. Like, see, in that, basically, we use calcium chloride. And calcium chloride sort of creates pores in the cell membrane, thus allowing the, you can say, the host cell to take up that DNA, that recombinant DNA in itself. And then it is placed on a cool ice bath. So it's like heat shock and then reducing the temperature. So that is what we mean by transformation. Then another method is called as microinjection. Now, microinjection is that process wherein the nucleus or the gene, the cell is held on in place with the help of a pipette, and then with the help of a microinjection, the gene of interest or the recombinant DNA is injected directly into the animal cell. This is specifically there for the animal cell. Then Another method is biolistics, which is particularly there for the plant cells mostly. How? What we do here, we take high velocity microparticles of gold, of tungsten, and then we bombard the cells with it, right? This is called as biolistics. You call it as gene gun. Furthermore, tumor-inducing agrobacterium. Now, Agrobacterium tumefaciens is a bacteria which causes crown gall diseases in the dicot plants. It has a natural mechanism of doing a lot of cell division. So if we take the pathogenicity of that TI plasmid and we use the TI plasmid to deliver the gene of interest by making it unarmed or disarmed, then you can easily transform the plant cell. And in the case of the animal cell, you can do the exact same thing with the help of retroviruses. Then another method is electroporation. Now, electroporation is when short electrical impulses of high field strength are given. This increases the permeability of the protoplasm membrane, and then the DNA molecule is taken inside the cell. So it increases the porosity, it increases the pores. Once you have done that, you have transformed the host cell. Now you culture the host cell for getting the product, right? Multiple copies of it. And that is done also you can say on an industrial scale. It is done by the fermenter. And the fermentation process is like the process wherein you use all the raw materials, you give it time, and then with the help of the various apparatus that is there, the impellers, the blades, the oxygen delivery and everything, it produces the product. Is it clear?
Yes, ma'am.
Any doubts? So with this, the biotechnology principles and processes chapter is done for the NEET session. Now, we will be solving more questions on this particular chapter so that we'll get used to, as to what kind of questions are asked. So you remember if we have solved the NCRT questions as well, isn't it? So we'll start with NCRT first. Because your NEET examination is going to be NCRT based only. So you must be clear with the NCRT, then only move on to any kind of reference book or any kind of reference questions. So we'll first do the biotechnology through NCRT and then we'll move on to different questions. So I'm sharing my screen. Tell me once you can see the screen that I'm sharing. Can you see the screen?
Yes, ma'am.
Okay, now you can solve these questions, then we'll move on to the next.
Done, ma'am. Done, ma'am. Done, ma'am.
Done, Jyobita? Jyobita, are you done till here? Hello, am I audible, Jyobita? Hello, am I audible, Jyobita? Can you hear me?
Yes, ma'am.
Are you done till here? Shall I scroll down?
Yes, yes. Done. Done, ma'am.
Done, Jyobita? Done till here?
Yes. Done.
Okay, Jyobita, any doubts in any of the questions?
No.
No doubts? All of the questions you were able to do so?
Yeah.
All right. Okay, so today we have completed the biotechnology principles and processes chapter. Now we will start with the applications part from the next class. Okay,
Okay,
so
ma'am.
let's just keep it till here only. We'll continue in the next class.
Okay, ma'am. Thank you.